Hi, and welcome back to my channel. In this video, we are going to be taking a look at DNA and DNA replication. This is a common topic that you look at in Biology 1, however, it is revisited in many other courses. I ask that if you enjoy this video and its contents, that you please make sure to like, share, subscribe, and turn on notifications so that you never miss out on a new video and so that I can continue making these videos for you. In this specific video, we are going to be looking at the history of DNA, the structure of DNA, and also DNA replication. DNA, or deoxyribonucleic acid, is a double helical structure that contains the hereditary material in living organisms. It is made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. But how did we come to find out this information? How did we find out that DNA is the hereditary material and that it's composed of certain things like the bases adenine, guanine, cytosine, and thymine? In order to understand this, we're going to delve a little bit deeper and look at some scientists that contributed to the finding of DNA. Now, there are many scientists who did contribute, and we're going to highlight some of the ones that contributed to this in the next few slides. In 1953, James Watson and Francis Crick introduced an elegant double helical model for the structure of deoxyribonucleic acid, or DNA. They are famous for their discovery of the first correct double helix model of DNA structure and eventually received the Nobel Prize in Physiology or Medicine along with Maurice Wilkins, another member of their lab. However, there are many other scientists who conducted DNA research prior to Watson and Crick, and their discoveries led to our ability to understand DNA as we know it now. In the next few slides, I will highlight a couple of them and their work, and eventually come back to Watson and Crick and their discoveries. The discovery of the genetic role of DNA began with research by Frederick Griffith in 1928. Griffith worked with 2 strains of Streptococcus pneumoniae, which are bacterium. One was pathogenic and one was harmless. The S strain was so called because this specific bacteria, when it grew, formed colonies that were rounded and smooth, which is why it has the S abbreviation. The appearance of these bacteria is caused by a polysaccharide or sugar-based coat that is produced by these specific bacteria. The coat that is produced by this bacteria actually protects the bacteria so that when it enters into the body, it is not recognized by the immune system. So what he did was he took these living S cells and he injected them into a mouse. He did his experiments on mice, and when he injected these S cells into the mouse, he found out that the mouse got pneumonia and eventually died. And that's because this coat that is on these specific bacteria protects these bacteria, and when it is injected, the immune system of the mouse does not recognize it. They can continue to grow and cause disease. So this specific S strain is the virulent strain, or the strain that could cause disease. The R strain, when grown in culture, formed colonies that had well-defined edges and a rough appearance. This is why it is given the R abbreviation. In the case of these bacteria, they do not have a coat like the S strain, and therefore they could not hide from the immune system of the mouse. So when these were injected into the mouse stayed healthy because the immune system was able to recognize these bacteria without the coat and clear them from the mouse's system before it was able to grow and cause any type of disease. So the R cells were non-pathogenic, and this was the control. So when he injected those into the mice, those specific mice stayed healthy. He then went further to take heat-killed S cells, those pathogenic ones. He heat-killed them, and then he injected them into the mouse. And just like he suspected, he had killed the ability of these bacteria cells to grow multiply, and cause disease. So when these heat-killed S cells were injected into a mouse, the mouse stayed healthy. The final thing he looked at was taking a mixture of those heat-killed S cells, like he did in the previous experiment, but this time he mixed them with living R cells. Remember, those non-pathogenic control cells. And when he injected these into the mouse, he found that the mouse would develop pneumonia and die. Not only did he find out that the mouse would develop pneumonia and die, but when he took blood from these specific mice, he found that they still contained living S cell bacteria. From these experiments, he concluded that the R strain bacteria must have taken up what he called a transforming principle from these heat-killed S cell bacteria. This allowed the R strain bacteria to transform into the virulent S-coated smooth bacteria. We now know this phenomenon as transformation, and the transforming principle that he referred to, we now know that as DNA, and that DNA can be transferred over to other bacteria and expressed as we see in his experiment. The transforming principle was later confirmed by experiments done by Avery, McLeod, McCarthy, as well as experiments by Hershey and Chase. In 1944, three scientists, Avery, McCarthy, and McLeod, did a series of experiments to determine what Griffith's transforming principle was. In their experiments, they began with large cultures of heat-killed S cells, the same cells that Griffith had used. In a series of steps, they purified the transforming principle by washing away, separating out, or enzymatically destroying the other components that were there. Eventually, they were able to purify the transforming principle. They were then able to analyze this and determine that the transforming principle might be DNA. There were several results from their experiments that led them to this idea. One of them was that the purified substance that they got gave a negative result in tests known to detect proteins, but a very strong positive result in tests known to detect DNA. Through other tests, they found that the composition of the purified transforming principle very closely resembled DNA in its ratio of nitrogen and phosphorus. Finally, they found in their experiments that RNA and protein degrading enzymes had little effect on this transforming principle, but enzymes that they used that degraded DNA eliminated the transforming activity altogether. Even though all this data suggested that DNA might be the transforming principle, Avery was very cautious in interpreting his results. He thought that it could be possible that his purified transforming principle wasn't all the way purified and there may be something else contaminating it. Because of this, there was much debate over DNA's role until about 1952 when Alfred Hershey and Martha Chase used a different approach to identify DNA as the genetic material. Hershey and Chase used bacteriophages, which are viruses that attack bacteria. They used radioactive sulfur and phosphorus to trace the fates of protein and DNA of the bacteriophages that infected the bacteria cells. They used two different batches in order to show this. In batch 1, the phages were grown with radioactive sulfur, which was incorporated into the protein portion of the phage. In batch 2, the phages were grown with radioactive phosphorus, which was incorporated into the DNA portion of the phage. After they grew up these phages, they mixed these phages with bacteria, and the phages were able to infect the bacterial cells. What they found was that the proteins that were labeled with radioactivity remained outside the cells, but when the DNA was labeled with the radioactivity, it was found inside of the cells. This showed that the DNA had actually entered into the bacterial cells, but the proteins did not. Hershey and Chase then concluded that DNA, not protein, functions as the genetic material, which finally solidified the work of Griffith in that the transforming principle was indeed DNA. It was known that DNA is a polymer of nucleotides, each consisting of a nitrogenous base, a sugar, and a phosphate group. In 1950, Chargaff reported that DNA composition varies from one species to the next. This evidence of diversity made DNA a more credible candidate for the genetic material. Two findings became known as Chargaff's rules. One, the base composition of DNA varies between species. His second rule states that the number of adenine and thymine bases are equal, and that the number of guanine and cytosine bases are equal. This second rule was a very important finding because it helped Watson and Crick determine the double helix structure of DNA. After DNA was finally accepted as the genetic material, the challenge was to determine how its structure accounts for its role in heredity. Maurice Wilkins and Rosalind Franklin were using a technique called X-ray crystallography to study the molecular structure. Franklin produced a picture of the DNA molecule using this technique. Franklin's X-ray crystallographic images of DNA enabled Watson to deduce that DNA was helical. The X-ray images also enabled Watson to deduce the width of the helix and the spacing of the nitrogenous bases. The pattern in the photo suggested that the DNA molecule was made up of two strands, forming a double helix. Both Watson and Crick built models of a double helix to conform to the X-rays and the chemistry of DNA. Watson built a model in which the backbones were antiparallel, meaning that their subunits run in opposite directions. Watson and Crick determined that the adenine paired only with the thymine and that the guanine only paired with the cytosine. The Watson-Crick model explains Chargaff's rules. In any organisms, the amount of adenine equals thymine and the amount of guanine equals cytosine. In this figure, we see the key features of DNA structure. DNA is a double helix in which the adenine binds to a thymine and cytosines bind to guanine. The nucleotides consist of a nitrogenous base, a sugar, and a phosphate group. The nucleotides are bound together by hydrogen bonds. But how does DNA then replicate itself? The idea was that the hydrogen bonds would break and that the two chains would separate from each other, and then the chain would act as a template. But there were three different models in which that could occur. The first one was the conservative model. In the conservative model, the parental strands come back together after they both separate and are used as a template, so that the parent strands will come together and then the daughter strands would also come together. In the semi-conservative model, the parent strands would be used as the template, but they would reconfigure so that the parent strands would remain with one daughter strand each. In the final model, the dispersive model, each strand of both daughter molecules contains a mixture of old and newly synthesized DNA. Watson and Crick's model predicted that the semi-conservative model was the way in which DNA replicated. This is again when the double helix replicates and opens, each daughter molecule will have one old strand and one new strand. Eventually, experiments conducted by Matthew Meselson and Franklin Stahl supported the semi-conservative model of DNA replication that was predicted by Watson and Crick. Let's talk about DNA replication. What exactly is DNA replication? DNA replication is when DNA replicates itself so that a cell can make a copy of itself. Now, DNA replication requires the copying of the entire strand of DNA. This uses a lot of resources and energy, and so the cell is not going to do this just for fun. The cell will replicate only when the cell itself needs to divide. Now, before we start talking about DNA replication and how that happens, let's talk a little bit about the orientation of DNA. DNA is a double helical structure that has two strands. One runs in the 5' to 3' direction, and the other one runs anti-parallel to that. Now, what do the 5' and 3' numbers indicate? These indicate the carbon numbers in the DNA's sugar backbone. The 5' carbon has a phosphate group attached to it, and the 3' carbon has a hydroxyl group. This asymmetry gives a DNA strand a direction. DNA is normally read in the 5' to 3' manner. And when we talk about DNA polymerase is going to work in a 5' to 3' direction. Another thing to understand about DNA replication is that, like I just said before, DNA replication is semiconservative, so that each strand in the double helix is going to act as a template for synthesis of the new strand. We are going to go ahead and take a look at how DNA replicates and the key players that are involved in its replication. So how does DNA replication happen? Remember, between the nucleotides, there are hydrogen bonds that are holding them together. So the hydrogen bonds aren't very strong, and they can easily be broken. So we see an enzyme called helicases, and helicases are enzymes that are needed because they untwist the double helix, and they open up the two strands from one another so that replication can take place. So basically, because of the helicases, these hydrogen bonds can break open and allow these strands to be copied. And I'm going to show you how that happens. Another thing that's important is that this does not happen in the entire DNA all at once. It happens in little places. And so these little places that it happens in are referred to as origins of replication, or sometimes they are what is called a replication bubble because they open up into a bubble. So if we take a closer look at the two strands of the DNA that I've drawn here on its side, after helicase opens it up, it forms this area here referred to as the replication bubble. And like I mentioned before, so this thing would be the replication bubble that's opening up here while everything is still closed on the sides. And then the area where it opened up is referred to as the origin of replication. Now, this is going to keep happening throughout the DNA strand where it's going to open up. The strands will then be replicated, and then they will close up, and then it will continue opening up and closing up. And that way the entire DNA is not all open at the same time. Another terminology to look at is that this area right here that is opening up is referred to as the replication fork. So we're going to continue to open up in this direction, and right before the replication fork there's an enzyme that sits right before it, and that enzyme is called topoisomerase. And this enzyme is really important because its purpose is to help relieve the strain as the DNA is opening up behind it. It helps to relieve the strain that is occurring in the DNA itself by breaking, swiveling, and rejoining DNA strands. It helps to relieve that tension that's going to be created there from not allowing the DNA to get too tightly wound as it's opening up. So now that we have the replication bubble opened up by the helicase enzyme, let's take a look at how the replication of the DNA occurs. The next enzyme that is going to be needed is primase. Primase synthesizes an RNA primer at the 5' end of what's going to be called the leading strand, and at the 5' end of each Okazaki fragment of the lagging strand. We'll talk a little bit more about what the leading strand and the lagging strand is, but for right now it's important to know that DNA, again, its correct orientation is from 5' to 3', and this is also how the polymerase makes DNA, is from 5' to 3'. So the primase is going to bind to this area, that's going to allow DNA to be made 5' to 3'. So it would bind to the 3' area here, and then it would actually bind closer to the replication fork on the other side. So on one end it's going to bind farther away from the replication fork. On the other end, they don't show it to you on the bottom piece right now on this picture, but on the bottom end it would bind closer to the replication fork. So again, this primase is going to attach here where the 5' end would start to be made. So on the 3' end here, where the 5' new strand is going to be made, and then over on this side, closer to the replication fork for the other side. This primase is necessary because it's going to lay down an RNA primer, and this is going to be important for our next enzyme that's going to play a role in this. So the next enzyme that we're going to see play a role here is DNA polymerase 3. And DNA polymerase 3 is going to start synthesizing the strand after the RNA primer is made. So it's going to recognize that primer position that was made first by that primase, and then DNA polymerase is going to continue synthesizing that DNA strand. The DNA polymerase will continue to add those nucleotides until the new strand is synthesized. Now, one thing to note here is that when it is working from this 3' end of its template, meaning it is laying down nucleotides in the 5' to 3' direction, this is going to continue continuously, as you can see that. So as Helicase keeps opening this up at the replication fork, DNA polymerase can continue to make this, and it will continue to make this continuously, meaning there's going to be no breaks in there. And that's what is called the leading strand. So I had mentioned the leading strand before, and this one is referred to as the leading strand of DNA because it's going to be formed continuously going in the 5' to 3' direction with no breaking up of the DNA. Now, what exactly is happening as the DNA polymerase is working? So kind of just for an example, if we quickly put this as this template up here, remember this is the template going in the 3' to the 5' direction, and then we just give it some nucleotides here. Okay, just for an example. Now this is the one that's being synthesized. Let's give it a different color. This one's being synthesized by the DNA polymerase. So what is a DNA polymerase? It's putting the nucleotides down that match up to the nucleotides on the template. So remember, the one I've drawn in black up here that I'm starring is the template, and then we're making our leading strand down here by putting down the nucleotides that match up to that template. So the DNA polymerase is responsible for putting these nucleotides down that are matching up, and that's why it's using this as its template, and it's going to continue on in this direction continuously to make that leading strand. But we also have the other piece of DNA that runs in the opposite direction, where it's running from 3' to 5'. And if we are opening up here, and they haven't shown you here, but this up here, this area right here would be the replication fork. Okay, just like in the other picture, they just haven't drawn everything here. So it's opening up here and continuing to opening up in that direction. Now again, DNA has to be made from the 5' to 3' direction, but the problem with this one is it's oriented in the opposite direction. because it's anti-parallel. And so what happens is every time it opens up, it gives that primase a chance to bind, and then the primase will then allow the DNA polymerase to bind. So it can only bind as far as where the replication fork is, and it has to go in the opposite direction. And so you see that here because it's going to be synthesized in the opposite direction. And so to continue that story, once the primase can bind, then again the DNA polymerase 3 can bind just like before, but this happens in little pieces because it cannot happen continuously. Once the replication fork opens again, then the RNAs, the primase can bind and make that RNA primer and then DNA polymerase. So you kind of see that this is going to happen in little chunks because it's opening backwards and going again and again. Okay, and so again, this is kind of the DNA, and then the RNA, the primase is going to bind here to make the primer, and it's going to go in this direction, and so then this is going to have to open up more, and then it's going to happen here. So you see that it's going to happen in little chunks as it occurs. And these little chunks that are made are called Okazaki fragments. So instead of having a continuous strand like we saw before, on the bottom portion we're going to have these Okazaki fragments. And so they're fragments of the DNA that's being made instead of continuous. And because of this, this is referred to as the lagging strand. So instead of being the leading strand, this is referred to as the lagging strand. Another enzyme that is going to play a role here is DNA polymerase 1. The purpose of DNA polymerase 1 is to remove the RNA nucleotides that were made by the primase and replace them with DNA nucleotides that are added to that 3' end. So now there will be no more RNA nucleotides, but only DNA nucleotides. The next thing that has to happen is we need to get rid of the Okazaki fragments. Remember, on the lagging strand, there's going to be those Okazaki fragments made because the replication fork can only open so much. And so there's going to be fragments made within that portion of the DNA, which is why it's called the lagging strand. In order to get rid of those fragments, DNA ligase is needed. And the purpose for DNA ligase is to join those Okazaki fragments together. Once the DNA ligase joins these Okazaki fragments, then the DNA strand will be whole. This table is a nice summary of all the enzymes that we went over that are involved in the replication of DNA. Once DNA replication is finished, you will have 2 new strands of double helix DNA, each one containing 1 parent strand, and that was used as the template, and then each one containing a new strand that was made off of the template. And that is called semiconservative DNA, where we're going to conserve the parent strand, use it as a template to make that new strand. So now, once it's completed, there will be 2 strands of DNA. After the DNA is made, there are DNA polymerases that proofread those newly made DNA to replace any incorrect nucleotides. If there are mismatched pairs of DNAs, then there are repair enzymes that can correct errors in the base pairing. The error rate after proofreading repair is low, but not zero. Sequence changes may become permanent and then can be passed on to the next generation. These changes or mutations are the source of genetic variation upon which natural selection operates. In the eukaryotic cell, DNA is combined with proteins in a complex called chromatin. The DNA itself, over here you see the double helix of DNA, is combined with these proteins called histones. These histones are responsible for the first level of DNA packaging in chromatin. They will bind together with the DNA and the DNA and wind it up very, very tightly. After the histones bind and start to wind it up tightly, once it's wound twice around this protein core, the histone protein core, it is referred to as a nucleosome, which looks like beads on a string. As it continues to pack, as the DNA continues to get packaged, it is referred to as a 30 nanometer fiber, as it is approximately 30 nanometers in thickness. Wrapping even further creates these loops in here where they look like loops, and that's why they are called looped domains. And then eventually it is wound into a chromosome, each individual pieces of this being a chromatid. We will talk more about chromosomes when we talk about mitosis. This concludes this video on DNA and DNA replication. If you have any questions, please don't hesitate to write them in the comments below. Thank you, and I hope you enjoyed this video.